as table sugar, and it is used as what as sweetener. It is found in sugarcane and also what beetles. That's actually one thing about what about sucrose. Now let's move to the next example. The next example that you can have is the case of what of you can have lactose. Lactose, which is commonly referred to as milk sugar. Yes, milk sugar. That is what lactose is what is called. But one thing about lactose is that it is formed from the condensation reaction that actually occur between one glucose residue and also what galactose. Galactose is a monosaccharide, a six carbon unit monosaccharide. It has a structural resemblance to that of glucose, but one thing is just that at carbon number 4, the orientation of the hydroxy group and also what the hydrogen group is actually different. So that's one thing. That's what makes the lactose be different from glucose. So this because of the orientation of the hydroxy group on carbon number 4. So lactose is formed from the condensation reaction that occur between glucose and also what galactose. And as you can see, everything that you are saying here, one glucose residue, one galactose residue, it shows that we are having two monosaccharides coming together to form what a disaccharide, right? So that's another example that of disaccharide that you can have. So another example that you can have in this case is actually the case of you can have maltose as well. Maltose. And one thing about maltose is that maltose is formed from the condensation reaction that occurs between two glucose residue. When you're having two glucose residue, right? That's the formation of maltose. But if I actually move to the details of maltose, let me just simply explain something about about lactose. Lactose is mostly referred to as milk sugar actually. But one thing about lactose is that it is like it promotes calcium absorption. That's one thing about lactose. It promotes calcium absorption. That is the rate at which calcium is being absorbed. Lactose actually promote it. But one thing, what I want you guys to understand is that lactose is basically source of food in infants. Yes, infant, that is small pikin. Yeah, so small child. That's what I mean. So lactose is an important source of food for them because their body have an enzyme that can actually metabolize lactose. The enzyme that can metabolize lactose. But in the case of adults, the activity of the enzyme that can metabolize lactose has actually decreases. So adults cannot really metabolize milk. Yes, so the milk that you guys are taking is actually digested milk. They've already digested, so you don't have anything to do than just to take it. But normal milk, infants, their enzyme is still very active. The enzyme that can break down lactose is still very active in infants, right? So that's just it. So lactose, whenever you condense them together, you're going to glucose and galactose. Whenever you condense them together, you're going to form lactose, and whenever you hydrolyze lactose, you're going to form glucose and also galactose. That's also actually what happened here. Now, in the case of maltose is formed from the reaction that occurs between glucose and also glucose, two glucose residue, right? So, and one thing about maltose as well is that maltose is commonly an energy intermediate. It is very important when it comes to digestion, starch digestion actually, because whenever you're talking about starch is actually a polysaccharide that contain, or let me just say homopolysaccharide, because it contain glucose all through. So the hydrolysis of starch, or let me simply say hydrolysis of starch, will yield maltose on digestion. Whenever you're talking about digestion of food, maltose is actually is actually being produced, whereby maltase, an enzyme that acts on maltose, now break it down to glucose, two glucose residue. So one thing about maltose is that it's actually a very let me just simply say an energy intermediate when it comes to digestion. So that's it. That shows that the function of maltose is actually maltose actually function in energy generation. That's what you can actually attach to it. That's its function. Energy generation. Exactly. That's function of maltose. Now let's go ahead and talk about the other ones that we have. The other disaccharide that we have, right? Let's talk about that one as well, one by one. So let's move to the next one that we can have. The fourth one that we can have. You can have the case of trehalose, right? You can have the case of trehalose. Yeah, trehalose is actually not common. It's not really common like that. It's not really common. But one thing is that it has a structural resemblance to that of maltose. Yeah, its structure is somehow similar to that of maltose, but the orientation of the bond is different. Yes. the orientation of the bond in maltose is different from that of what, of trehalose. So that's what it is. Trehalose actually contain two glucose molecule, right, that are condensed together. So but the orientation of the bond is different. So once you get to the structural aspect of trehalose, I'm going to show you the orientation of the bond and also more about it. But just for listening very well, trehalose is actually found in what, in the structure of some insects. It is let me just say energy storage. Let me just use the word structure. It is energy storage in some insects, yeah, some insects, and also to some extent in plants as well. And they function as energy generation in fungi. Yes, energy generation is fungi. They are the storage form of energy storage in fungi. So that's just it. And some insects and plants to some extent. So that's just it. Another function is that they protect. They helps in they protect against dehydration. Let me use that word. They protect against dehydration. That is, they helps in what, in dehydration. That's just one thing about trehalose. So, and just know that they have structural resemblance to that of what, of maltose, right? And they are found in what, in some insects, fungi, and plants as energy storage. Yeah, let me add the word energy storage. They are found in some insects as energy storage, and they help in what, in dehydration. That's actually one of their function, and that's just it. Now another monosaccharide, which is the last one that we can have, is actually the one that is found whenever we are talking about digestion of food, which is the isomaltase. Isomaltase is also mostly referred to as what, as let me use the word alpha limit dextrin. Although one thing is that, let me just put it like this: once you hydrolyze alpha limit dextrin, alpha limit dextrin is actually an oligosaccharide. Yeah, it's an oligosaccharide. Alpha limit dextrin is an oligosaccharide that contain alpha 1,4 glycosidic bond. It contains alpha 1,4 glycosidic bond, and at the same time it contains alpha 1,6 glycosidic bond. So it is called alpha limit dextrin because once you start breaking the bond of starch, you break it to some extent, you are going to reach alpha 1,6, right? And at the same time, you are going to reach what, alpha 1,4. One thing is that after our body really have an enzyme that can actually act on alpha 1,4 glycosidic bond, but not really that can act on alpha 1,6 glycosidic bond. But what actually happens is that there will be a change in the orientation of alpha 1,4 glycosidic bond to alpha 1,4. There will be a change in alpha 1,6 to alpha 1,4, and that's actually happen whenever we are talking about glycogenolysis. That is breaking down of glycogen in the body. Yes, there are some enzymes that actually acted by transferring the position of this alpha 1,6 to alpha 1,4. But once you get to glycogenolysis, because I'm still going to explain the metabolism aspects of this carbohydrate. So once you get to that glycogenolysis, I'm going to explain how the enzyme actually work, how they basically convert alpha 1,4 glycosidic bond to alpha 1,6 glycosidic bond to alpha 1,4. Because our body can actually it actually has an enzyme that can act on this alpha 1,4, but not really that can act on alpha 1,6. That's why you have alpha limit dextrin, which is later acted on by an enzyme that we called isomaltase. So that's just it. So I'm going to explain isomaltose. It's a type of disaccharide that we actually have, a types of disaccharide as well. So it's also and let me just say intermediate of energy generation, like of energy. Whenever your body is metabolizing your carbohydrates, alpha limit dextrin is actually an oligosaccharide. At the same time, isomaltose is also a disaccharide, right? So that shows that it's composed of what? Of glucose and also what? Glucose. But one thing that happens here is that the orientation of the glucose is different from that of what? Of maltose. Don't forget, isomaltose, we have maltose, and also at the same time we have what? We have trehalose. They are actually disaccharides that contain what? Glucose residue. But one thing that happens here is that their orientation of the bond is different. Yes, the orientation of the bond is different. One is alpha 1,4, one is alpha 1 6 upon 1 2. That's actually how it's happened. So their orientation is actually very, very different. But once you get to the structure of disaccharides, I'm going to explain how they join together, how triallose is formed, the structure of the triallose, the bond, the type of bond that is found in the structure of triallose. I'm going to explain it, and that was going to be in a separate video. Now let's just go. So in the case of isomaltose, is actually glucose, contain two glucose residue, right? And one thing is that the bond that are found in isomaltose is different from that of maltose itself. That's why they use the word isomaltose. Now from there, the function is that it's actually an intermediate. It's an intermediate in starch digestion. Yes, let's use the word starch digestion. That's just it. So it's an intermediate in starch digestion. Whenever you are let's say your body is let me just say is digesting something like rice now. No, rice is actually a starchy food. So once your body is metabolizing starch, you're going to have the nature of isomaltase. It is isomaltose. It is very, very, very confirmed. Check it now. So isomaltose is actually an intermediate. That is, it is formed in between starch digestion. You get it down. It is formed in between starch digestion, and also it is formed to some extent in some microorganisms as well. To some extent in some microorganisms, right? Microorganisms, right? So it is found in to some extent in microorganisms. That's one thing, right? So another thing I want you guys to know is that to some extent as well, it is found in some food. Exactly, it is found in some food. So that just is about isomaltase. So all these disaccharides they are talking about, they are actually very, very important when it comes to energy generation. Every one of them are very, very important in energy generation. So I'm going to summarize their function out. So they are very, very important when it comes to energy generation. That's the first thing. Energy generation. Energy generation.